Well, it looks like we're getting a number of people popping in. I'll give it one more minute. Just give me a sec here. I'm having a little bit of Uh, okay, good morning, everyone, and good afternoon, depending on where you are. So I think we can get started. There's a fair number of people on the meeting now. I did send out the agenda prior to this meeting, so just to quickly run through, Sophia will give a brief update on the status of the M4K website. I'll turn things over to Charles River, and we'll give an update on a proposal for a rat DMPK study and some background on some work being done to publish some work on PROTAC inhibitors for ALK2. Then we'll move into the lead compounds for development. The ICR will give an update on some recent DIPG patient-derived cell data. We'll move into the table describing what we're going to do to fill in the gap for data on 2308 and the scale up, and then we'll finish off with some updates on some grants that we've been applying for and some funding opportunities. So Sophia, if you want to please just give a brief update on the website.
Thank you, Peter. Good morning, everyone. Not much to report here. We're trying to update the website with updated logos and information that are kind of outdated. So if you have noticed anything, please let me know, as we are in the process of doing that within the next month. We are hoping to have kind of resource sub webpage within the website that can highlight some of this meeting data that we want to share in public. But other than that, I think it's a work in progress for now, so too early to report anything really important.
Okay, great. Thank you, Sophia. So we can move on to Charles River, and I believe Divya, are you going to give this update?
Yep. Yeah, that'll
okay.
be from me.
Great.
Yep. Okay, so just to give a bit of context to the study being proposed, so we brain levels were detected for 2308 in mice following the dosing of its methylated parent 2281. As 2308 has improved cellular potency in the nanobret assay compared to 2009, it was thought to have a therapeutic advantage over 2009. However, with the DIPG cell assay run in Chris Jones's lab, this showed the compounds to be equipotent based on the preliminary data. So it was deemed important to have the actual brain data for both compounds in an equivalent set of conditions and to determine which candidate is the most developable. So in terms of the study design that we're proposing, we would dose 2308 in a full PK study in SD rat, and the study design would be the same as what was carried out for 2009 and at Pharmaron. And this would allow us to get an understanding of the developability of this compound and to get a IV in vivo in vitro in vivo correlation of the clearance using the microsomal data that we already have. But ideally, we would also need to get hepatocyte stability data for these compounds as well. Alongside this, we would dose both compounds orally with terminal samples taken at 3 time points, which are outlined up here. This would give us more of an insight into the distribution of the compound when taking the blood, whether it's distributed into red blood cells and then also into muscle, which would allow us to have an understanding where there is no little or no efflux transporter activity there as well. And then also, and most importantly, to be able to get the brain data for both compounds to be able to estimate target receptor occupancy. We have proposed dose levels of 1 mg per kg IV and 5 mg per kg PO for the study. However, this would be finalized once the data from the mouse brain distribution study has been completed over in OICR. I've also made an estimation on the compound requirements based on the higher oral dose of 10 mg per kg. And the cost of this study would be approximately $26,000 US. Following the brain distribution study, we would the mouse brain distribution study, we would decide on whether to go ahead with the side-by-side brain distribution study. We would recommend that whether we see good or poor distribution in the mouse, this rat PK study would give us data in a second rodent species, and this rodent species would also be one that is used in tox studies as well. So in terms of recommendations, we would like to proceed into rat PK studies as outlined above, and also to confirm preclinical toxicology species selection for pre-candidate compound following the cross-species metabolite identification, so MET-ID study. That's it from this slide. If anyone's got any questions.
Yeah, thanks, Divya. Any questions for Divya? Okay, yeah, we should have that data on the mouse study coming up by the end of the month, I would think.
Okay.
Okay, so if there's no questions for Divya, we can move on to some PROTAC work.
Yes. I don't know if Dan's on the line. He's going to cover this.
Sure, thanks. Hi, everyone. I'm Dan. I'm a PhD student here at Charles River. I'm just I wanted to talk about where we're kind of in the very early stages of thinking about doing a publication on all of the PROTAC work that I've been working on and that we've been doing here at Charles River. And we're now in the process of getting together all of the data that we've got and all of the data that we might want to include in order to prepare a publication. And so the kind of rough outline that we've got at the minute is that we want to write a chemical biology style paper, not looking at a particular disease, but looking in general at ALK2 degraders and then all their profiling in cells. And so kind of our pathway here would be we would introduce BMP signaling and the role that it has in various diseases, FOP, diffuse midline glioma and glioblastoma as well. We would discuss kind of the conversion of ALK2 inhibitors like M4K2009 into a kind of first generation ALK2 PROTACs, and a couple of example compounds are shown in the bottom left of the slide, compounds 2 and 3. And then we talk about how we profiled these compounds and evaluated them as hits using nanobret assay, which was run at UNC, and also Western blot ALK2 degradation data we've done here at Charles River, and also we've generated a whole load of in vitro ADME data on a lot of these PROTACs, which is really interesting. We'd then kind of talk about the optimization of these first generation PROTACs, where we kind of rigidified within a linker portion, and one of our best compounds here was M4K2308. and kind of talk about how we developed this compound, how we prepared an associated negative control, and again, ALK2 engagement in cells, ALK2 degradation, in vitro ADME data, particularly focusing on metabolic stability of this compound, which we got a significant improvement from by changing the linker. And then also looking at a load of selectivity studies, particularly using proteomics. So we've got proteomics done in hex cells, looking at transfected hex cells with ACVR1, and we've also now got proteomics going on in a second cell line which endogenously expresses ALK2. We then really would like to focus in detail on kind of the mechanistic studies that we did around these compounds, really looking at the structural basis for ALK2 degradation using modeling and biophysical assays and things like that. And then also looking at the actual mechanism of how ALK2 is ubiquitinated and then degraded via a combination of a proteasome and a lysosome. And that's something we really want to focus on. And then to finish, we kind of want to show some sort of downstream effect, showing what ALK2 degradation actually does in sensitive cells. And so we're looking at a number of different options here, looking at potentially the patient-derived DIPG cell data from the ICR, but also we're looking at glioblastoma cells that express ALK2 and that are sensitive to bone morphogenetic protein signaling. And all of those studies are kind of in progress now. But that's a rough idea for what we're thinking.
And at this point, you have no plans to do any further synthesis? The synthesis part of this has wrapped up?
The synthesis part hasn't. We've made a whole iteration of new compounds, but our thinking was that that could all be contained in a subsequent paper where we focus on the optimization of our PROTACs for a particular disease order and disease, and also looking at trying to get CNS penetrant PROTACs. But that's all. That would all be contained in a subsequent piece of work.
And all of those new compounds, they haven't really been tested too much at this point, so all of that work would still need to be done.
Yep.
Okay. Any questions for Dan? Okay, thank you, Dan.
No problem.
Moving on. So up next, we'll turn it over to Rebecca, and we'll give an update on some recent cell line data.
Hi everyone. Yeah, so there's a few slides that all look quite similar. On the left-hand side will be the curves that we've generated, the dose response curves. Anything that's purple is ACVR1 mutant, and anything that's grey is ACVR1 wild type, but they're all diffuse midline glioma. The very bottom model in black, the MO3, is one of our normal control lines. That's only been run once. That's one biological replica that we're showing here. Everything else is 3. And then on the right-hand side is just a summary of the GI50s that we've determined from these curves. So for 2009 and 2308, which are currently the lead candidates, in our hands, the GI50s are very comparable. We don't see an increased potency with 2308 that perhaps you might have been expecting from the assay data that you had already. And then as we kind of move through the slides, if that's okay, Peter.
Sure.
It's just the other compounds that were also sent to us. So these numbers probably mean a little bit more to you in terms of their structures and where they've come from. These two have slightly higher GI50s, but really all the compounds that we tested were in the same kind of ballpark, so sub kind of 10 micromolar. There was only one compound, which I think might, if you move forward.
2163.
Yeah, so that's one that we've all previously tested in the lab before and more potent, and that's kind of similar to what we see in the data that we had with the compound before you sent this one through. Also to note that in most of these you do see a difference in the ACVR1 mutants being more sensitive to the compounds than the ACVR1 wild type, but the caveat that there's only 2 wild types here at the minute. So as far as stats, we haven't run it because the numbers aren't quite there yet, but we can top that up with an extra wild type line if we want to get some stats out of this.
Mm-hmm.
And then moving forward again, I think this was the one that actually, so the 2306 was probably the least potent that we've seen out of all of the ones that got sent. A lot of the GI50 curves not really coming down past our top concentration of 30 micromolar. The 3159 kind of around the ballpark of 10 micromolar. And then I think on the final slide, oh no, sorry, one more compound. Sorry to be whizzing through these if you're making notes, but on the next slide, it's us comparing GI50 to their end of the curve. And I don't think that's strictly necessary here, just because the curves generally fit quite well. They've got a top, a middle, and the bottom, so the GI50s are quite reliable. But on some of the curves where you're not hitting that 50% mark, we've included the area under the curve as well, just so we can compare. But the bottom line of this being that the patterns or the trends map whether you're looking at GI50 or area under the curve. But we can also share the raw data and values and anything you need for this.
Yeah, the values.
Just to point out, sorry, this was all Jiin. You can give away Jiin. Sorry, I'm doing all the chatting. This was all Jiin's hard work. So yeah, thanks to her.
Oh yeah, thanks. Yeah, so please send the values and the data, and I can incorporate those into the table. I did have one question about the mutant line DIPG 36. It seems across the board that's not a very sensitive cell line, at least for our compounds. In other, I guess, assays that you've tested or different compounds, whether they're ALK2 or not, how does that cell line, how sensitive is that cell line to other therapeutics that you've tested?
Yeah, so well spotted. And yes, we have got a couple of lines that are mutant and wild type and kind of behave in a way that we don't want it to expect them to behave. With 36, it's not always the least sensitive to every compound we've tested.
Okay.
There's an ACVR1 wild type model, the DIPG6, always there, always tends to be a bit more sensitive than the other wild type models. And we try and include all of these when we do our big panels, so that we're not just picking the ones that we know aren't going to respond, the ones that do. And that's why you end up with that variation. But it's not that DIPG36 just never responds to drugs. It's just that it seems pretty consistent here with these ALK2 inhibitors of potentially similar structures, that it's just not as sensitive.
Right. Yeah. So other ALK2 inhibitors that you've tested, they respond well, or?
Yeah, I guess with the caveat that some of the other two ALK2 inhibitors that we've tested aren't particularly selective.
Right.
Right. So yeah.
Yeah. So I think based on this data, that 2308 is essentially equipotent to 2009. I would suggest, unless anyone has any other thoughts otherwise, that we do move that into the in vivo efficacy model. And I'm wondering which model would you suggest?
So in vivo work.
Yeah.
We've got a couple that we are trying to run a bit more routinely. We've got a patient-derived model, HSJD DIPG 007. We're currently testing a new stock of, or going to be testing a new stock of these cells to try and get the median survival to be a bit lower. Currently, in our hands, the median survival is around 200 days, so it's quite a long in vivo experiment. We do also have a mouse model which has a much shorter median survival, around 40 or 50 days. And probably Chris might have different suggestion. My gut feeling would be to go with that one first and just compare 2009 to 2308 in a shorter model. It's got ACVR1 mutation, it's a bit more controlled, it's not patient-derived. But it should give us an answer, hopefully. And with the amount of drug that you've initially scaled up, we could make that work. The issue with the very long in vivo efficacy studies is that we probably need more drug. But given this is all on the assumption that if we do some tolerability studies, it's comparable to M4K. The 2009, all my calculations are based on about 50 mg per kg But if for some reason we can get away with dosing less, then obviously that scales down how much we need to run the assays. But usually we go with the maximum tolerated dose.
Right.
But we have to do that testing in our hands as well.
Okay. So you would suggest that, yeah, we do rerun 2009 in this 40 to 50 day model?
Yeah, so we've got
okay. Yeah. And I think the compound requirement for that was about 3 grams?
Yeah, that would get us through with some tolerability and PK and the efficacy in the shorter model.
Okay.
The other question was as well, if we do run 2009, we'd also need around 3 grams of that, and whether you'd be able to supply that or whether it needs to be bought in. That's just something I wanted to raise.
Yeah, we do have workable quantities of 2009 still, so that shouldn't be an issue.
Sorry, could I ask a question? Do you have an awareness? Do we know like the transporter expressions of these cell lines? Because I'm just wondering if it's a permeability thing in terms of 2306, because obviously that was quite active, but we don't see an effect of these cells. And I'm just wondering if it's permeability and whether the differences between 2009 and 2308 could be due to PGP. I don't know if you have a.
We could have a look. Definitely got expression data.
But we haven't expressly gone and quantitated that across all our lines yet.
No.
But we could look at it for gene expression at least.
Yeah.
Yeah, just wondering if that could account for the reason why we're not seeing 2308 being better than 2009.
Yeah, we can have a look.
Okay, great, thank you.
Okay, any additional questions for the ICR team? Okay, well then I will move along to the scale up. So I did mention early on that we have completed the initial scale up of 5 grams of 2308. Those yields are represented in pink. I would say overall, we're not seeing any major difference in yields on scale up at Piramal. That wasn't really the purpose of this. The purpose is just to get material in hand, although I would have hoped that there might be some improvement in the yields of this synthesis. So that was delivered last week, and we do have another batch ongoing, and that's going to be a 10-gram batch. So far, everything is kind of tracking along nicely with what we've seen in the past. We're at this point right now. That compound is in purification, and we're expecting to have that material delivered mid-November. So we should have workable quantities of 2308 to complete, a number of those key studies coming up in the next couple of months. Any questions on scale up? Okay.
Sorry, yeah. Have you got anyone sort of looking at the route and improving chemistry at all?
So yeah, that's a great question, and I have gotten a couple of quotes on route scouting. And if I compare the quotes from offshoring to North American prices, the North American prices are on the order of 5 to 6 times more than what we're seeing if we were to do the work overseas, which I guess isn't too surprising, but usually I see a 3 to 4-fold difference, and this seems to be much greater. And a lot of it comes down to the cost of the raw materials, particularly if we need to get away from that catalyst. So there is a couple of potential things that we can look at remove, this particular catalyst in particular, I think is the one that's probably the most expensive.
I guess you also don't want a palladium catalyst right at the final step.
Yeah.
Ideally.
Ideally, you wouldn't want that. So ultimately, if we do move forward with 2308, then there will have to be a decision point of how much do we want to spend on scale up of this compound. So if we were to basically go through with this synthesis, yeah, it's going to be very expensive, which is why I would stress that we need to do that root scouting exercise. Once we're in a position to choose this compound for development, that would be basically a critical factor in determining, can we shorten the route of synthesis, remove some of those catalysts. Another strategy that I'd be looking at is if we do want to do majority of the work in North America, would be to at least do some of the early steps overseas to generate, say up to 2 or 3 steps before the final product, and then move that to North America. So that would save at least some cost on the early stage intermediates. For 2009, I don't think this, it would be there wouldn't be a lot of advantage to doing a root scouting. The synthesis is pretty straightforward as it is.
I don't think you get it much shorter than it is.
Yeah, and much cheaper. Okay. So now that we have that material in-house, we have shipped out some quantities of 2308 to Pharmaron to complete some of those experiments that we currently don't have data for. So, in progress, we will have CYP inhibition, AIMS, and dose tolerability. The compound is currently on its way to China, so I would expect those experiments hopefully to start next week or early the week after, and then data will start trickling in the next 2 or 3 weeks. The mouse brain plasma experiment to look at the unbound brain to plasma ratio, that's currently underway at the OICR. So we'll look forward to seeing that data, and as Divya presented earlier, that will be key to kind of informing that rat study. A few tests that I haven't pulled the trigger on yet, that would be the cell data in different mutant cell lines. We can have that run at Reaction Biology. And then, we did touch on the DIPG model. So I think once we have that dose tolerability, now that we have the cell data, we can pull the trigger on that study. I have been speaking with Eileen Shore's lab at UPenn about the FOP model. That the contract is still with the tech transfer office, so we're still waiting on that before we can pull the trigger on this model. Does anyone have any questions on where we are with compound progression? Okay, well, I think this is probably going to be a fairly short meeting then. So up next is funding and partner opportunities. So appreciate everyone's input for some of these grants. I know that some of it went down to the wire, but in terms of the DMOS grant. We were able to get that in, and the LOI was accepted. So we're in the process now of filling in everything we need for the full submission, which is due in about 4 weeks. And this will cover up to 33% eligible costs up to $1 million Canadian. So I have been talking to some Canadian partners to get quotes for some work that could be done in Canada, particularly with the Charles River team. We do have some quotes, and they would conceivably do the tox study at their Montreal facility. So provided the cost is it's cost effective to work there within the constraints of this grant, then you know, we do have some options that we can run some of this work in Canada. A second grant that we submitted the LOI to just a couple of weeks ago through the OICR, and that was, yeah, as mentioned, was submitted through the OICR. So we got that in on October 7th. We should hear back on the status of the LOI around the end of November. And, you know, if we're successful, then I think we'll be in good shape, because essentially everything that we've put together for the Conscience DMOS grant will essentially be able to copy and paste into the Cure Search grant. And to end off with some really good news, the Drury Lab was awarded a Cure Starts Now grant last week. So, you know, I know David is not on the line right now, but just wanted to congratulate him on being successful in getting that award. And so now he has some funding to be able to continue on supporting this ALK2 program in his lab. Does anyone have any comments or questions on the funding or partnering opportunities? Okay, I mean, that was essentially everything that I wanted to cover today in terms of the slide deck, but there's a couple of, I guess, items that I just wanted to discuss with some people. I know that the Charles River group, particularly Divya and Kim, have to drop off at the bottom of the hour on Wednesdays now. So if there are no objections, I'd like to move this meeting to the last Tuesday of the month at the same time. We don't have to make that decision today, but if anyone on the line has, you know, is unable to make that meeting, if it was on a Tuesday at the same time, please let me know. Otherwise, I'd like to, you know, switch that in order to accommodate, you know, particularly Kim for these meetings. And the second item is so there will not be a November team meeting for the wide group as we have it right now, the scientific team. That will be a steering committee meeting. The last, I guess, Tuesday or Wednesday of December is obviously Christmas, so I will schedule a team meeting the week before Christmas. I realize a lot of people might be taking vacation at that time, but you know, I will schedule that meeting, and if you can make it, great. If not, you know, we'll see you in the new year. Does anyone have any comments?
Oh, great meeting.
Okay. Well, thanks everyone.
Short one today.
Yeah, it was a short one today. But I think with certainly the next one, once we have a lot of that data coming in, particularly from Pharmamal, then we'll have a lot more to discuss.
Yeah. Hopefully, we'd also have the BBB from mice at the
Right.
time. So, yeah. Okay.
So, yeah, so it's taken a little bit of time to get to this point, but now that we have compound I think the data is going to start, trickling in.
Great. Thank you.
Thanks, Peter.
All right.
Yeah.
Thanks, everyone.
Right.
Bye.
Thanks.
Yes.
We'll talk to you soon. Bye.